Welcome to Citizen Science, stories of science we can do together, also known as the SciStarter podcast. In this episode, we're delving into the secret sex lives of bees and flowers, a World Turtle Day celebration, and a new weapon to clean up our nation's waterways, tampons.
So like around the edges here, that's just thick cotton.
Perhaps the most unlikely love story in the history of the world has been developing over the past 150 million years or so, and it's up to us, citizen scientists, to figure out the details. Flowers and bees were made for each other, literally. We wouldn't have one without the other. Flowers evolved to lure bees to their reproductive organs and carry their pollen to other flowers so they can bear seeds. Bees are just vegetarian wasps, focusing their entire lives on being able to find the pollen and nectar that they need to survive. Over millions of years, flowers and bees have co-evolved all sorts of adaptations and strategies to optimize their relationship. Some flowers are promiscuous and happily attract and get pollinated by a variety of bees. Others are more demure, safeguarding their pollen and nectar behind tightly closed petals or even in secret chambers, accessible to only particular types of bees. Similarly, some bees will poke their proboscises into any flower in sight, while others seek out only the one perfect flower suited to them. The thing is, very little is known about these secret liaisons. There are 20,000 species of bees, 4,000 in North America alone, and many are tricky to identify. And it's not like they and their flower partners are conveniently located or easy to spy on. But we need to learn fast, because climate change, habitat loss, and invasive species are destroying these ancient relationships, threatening not only the particular bees and flowers involved, but entire ecosystems and also food systems that depend on them. This is critically important work. Wildlife biologist Sam Droege heads up the Native Bee Inventory and Monitoring Laboratory in Beltsville, Maryland, or B-Lab for short. He and his team are trying to reveal these bee-flower relationships.
In order to do that, the easiest thing is to have the plants right here in our research area so we can walk out the door, set up our survey techniques and programs and equipment, rather than have to drive somewhere and find out that someone had mowed down all the plants, or they had finished blooming, or they haven't started blooming, or it rains. So we're building out a set of research gardens in the area, and this is basically phase one, grow the plants.
They're repurposing dozens of enclosures that were once used to raise endangered whooping cranes to create what will essentially be bee flower dating sites with patches of every native flower imaginable. It's a monumental task, so he's enlisted the help of community volunteers, including gardeners, amateur scientists, or just nature lovers.
So we have so much capacity here that we have opened up our research area, and some of these pens have been designed and are now run completely by community members. So these community folks come in, we provide the space and the water, they provide all the schlepping, right? So they're transplanting things, they're moving things, they're creating rows of plants, they're weeding them. They do all the basic work. We just provide a place for them to do that doesn't have deer and has high security, like you have to gate in order to get in here.
The volunteer help is a necessity, but it's also a feature of the program, since the volunteers take what they've learned here, along with thousands of excess native plants, back to their communities.
We are walking the talk, right? Yes, we're developing all these techniques to study bees and to do a better job of conserving them, but we're actually engaged with the community, and these are everything from federal agencies down to individual homeowners to actually take some of the stuff that we have discovered and plant the native plants back out into these environments, which make a difference because bees are not redlining suburban areas or urban areas. They are simply flying until they find the right plant.
To get data back from all the home gardeners planting native flowers, Sam and graduate student Janan El Hefnawi created Ask a Bumblebee, a citizen science project you can find at SciStarter, where you record what sorts of bees are visiting your flowers and report back to the researchers. So we're going to try the Citizen Science Project Ask a Bumblebee, and I've got my data sheet here that I downloaded from their website, and I have Seek open on my phone. And the whole idea is you find an area with some native plants, then you go around with Seek and identify those plants, only the ones that are flowering, but try to get all of them, even the little ones that are near the ground, anything that has a blossom. And then for 30 minutes, you go around and record on your data sheet how many bees are visiting each flower. And if you don't know your bees very well, then what you do is just write carpenter bees or bumblebees, and that's all they're interested in. But if you can tell what kind of carpenter bee or what kind of bumblebee, then you get a different data sheet, and then you write in the species. I'm not that great at it, so I'm just going to do bumblebees versus carpenter bees for now. So I'm going to have a go at it. Okay, so I open Seek and record the flowers here. Indigo, Beard's Tongue or Penstemon, Blue Star, which is Ansomia, and Sweet Spire. And then they want me to count only bumblebees and carpenter bees. So bumblebees are big with fuzzy bottoms. Carpenter bees bigger and have shiny bottoms. All right, my 30 minutes are up. I've counted my bees. I will say that it can be a little difficult to tell one bee from another and avoid counting the same bee over and over again. But I did my best, and I'm going to go back to the computer, upload my data to Ask a Bumblebee, and hopefully it'll be useful for them. In addition to bees, May is also the month for World Turtle Day, and who doesn't love turtles? The problem is, many people love them to death. Turtles, and especially box tortoises, are in trouble because so many people grab them from the wild to take home as pets or to sell to other turtle lovers, which is awful because wild populations are threatened, and one way we know that is thanks to citizen scientists reporting turtle encounters to HerpMapper. It's a website and mobile app used by both herpetologists and non-scientists to monitor and track populations of turtles and other reptiles, as well as amphibians. Chris Smith is Director of Public Affairs at HerpMapper.
The purpose really is to collect observational data from across the world, so it's a global project. And then we get that information and we get it into the hands of conservation and research organizations. And so, amphibians and reptiles face many conservation challenges. And so we try to, bring data collected from a variety of different folks and put it into a single data repository and then share it back out with, again, the folks on the ground doing research and conservation to benefit the species.
Chris says that participating is easy. You just download the HerpMapper app to your mobile device or save the website to your computer and then take photos of any reptiles or amphibians you encounter, noting the location and date.
And that's kind of at the bare minimum all you need. Certainly there are a few more data fields that folks can collect as well. If you use the mobile app, it will collect the location for you automatically right on your device. You can take your photo or take an audio recording of a frog call, for example, right in the app. But if you're not wanting to use a mobile device, you can certainly go out into the field, use your normal camera or normal audio recording device, come back, sit down at your computer, and log your observations kind of after the fact on your computer as well. So we're pretty flexible on how we allow folks to contribute observations.
And while you can do the same thing on iNaturalist, Chris says there's an important difference between the two apps.
One thing that HerpMapper does a little bit different than iNaturalist, and this is one of those kind of pros and cons depending on one's perspective, is that we do not provide point location data publicly. And that's because a lot of our amphibians and reptiles are subject to things like poaching and habitat degradation by people going out and wanting to visit them and see them firsthand. And so we tend to kind of err on the side of protecting specific locations and only provide those data to the researchers and the conservation agencies directly. And so that's just one difference. And so folks may notice that if they kind of compare us with iNaturalist.
So generally speaking, iNaturalist is great for most plants and animals, but you might want to avoid sharing information on any species, turtle or otherwise, that will attract unwanted attention. And while HerpMapper itself doesn't help you identify species the way iNaturalist does, Chris says they also have another project that does.
It's called whattheherp.com, and so you can actually run a photo through this system and it'll spit out in terms of percentages of the probability of a species ID.
Finally, you might wonder if you should submit everything you see or if they're looking for targeted data.
We do have some special projects, so turtles being one of them. You see my sign in the background here. We do have quite a few researchers using turtle road-related data. But generally speaking, the HerpMapper project accepts data for all amphibians and reptiles, whether they're common species, rare species, or anything in between. We have folks using data to do kind of large-scale phenological studies, so the studies of when species are active, and they may be interested in common species in some of those studies. And then we certainly have lots of folks using some of rare species data to really inform conservation management of populations, things of that nature. And so really it's all welcome. There's value in common species data, there's value in rare species data, and everything in between.
You can find a collection of turtle projects at SciStarter. And if you love turtles or you'd just like to discover what's so cool about turtles, might I recommend these books by award-winning artist David Carroll. He's a MacArthur Genius Award winner, and he's not only painted turtle portraits for over 60 years, but he's perhaps the #1 turtle citizen scientist. And no matter how much you love turtles, I don't think anybody in the world loves turtles the way David Carroll loves turtles. For 60 years, he's been getting up at the crack of dawn and tracking and observing the turtles living in the wetlands of New Hampshire, and his detailed journals have been a goldmine for scientists. Finally, tampons. They're not just for exactly what tampons are just for. They're also sophisticated water quality monitors when paired up with a soda bottle and a black light. Generally, testing streams for disease-causing E. coli bacteria found in raw sewage requires a lot of expertise and expensive equipment. But Kelly Albus, research scientist at the Texas Water Resources Institute, is championing an ingenious citizen science-friendly method called tampling that uses tampons, or, as she's required to say when working in Texas schools, cotton on a string.
Because it's a cotton medium, and cotton is really useful for absorbing optical brighteners. And optical brighteners are chemicals, it's a whole suite of chemicals that are added to things like paper, toilet paper, toothpaste, soap, laundry detergent. They make whites look whiter.
If a stream contains optical brighteners, it must be contaminated with raw sewage.
And the cotton actually attracts the optical brighteners to some extent. So we can get them out after just a few hours, shine a black light on them, and if they glow blue, you have a positive.
Kelly's developed a tampling protocol that can be used by scout troops, in classrooms, community groups, and others. All you need, other than tampons, is a 365 nanometer blacklight flashlight that costs about $40. Oh, and some old plastic bottles.
You take your plastic water bottle and then you cut slits into the side of the water bottle so that the water can flow through. Then you take your bottle and your tampon, you unwrap it, and it has that handy little string, which is so helpful for us because you drop it right into the bottle and you loop that string over the edge of the bottle and turn that lid, and then your tampon is dangling inside its little nice plastic cage. Now you need to make sure that you really secure your tampon because it will move downstream in a storm. We've tied it to cinder blocks and it's moved miles. So you need to make sure it's attached to something very secure like a tree or a bridge, and if you have to use a rock, make sure it's a big one because especially in these storm events, water is powerful. So you want to make sure that you tie your bottle to something, drop it in the stream, and you leave it there for about 4 to 48 hours, and then you pull it back out of the stream. You can kind of squeeze it dry a little bit, take it back to the lab, unwrap it, shine your black light on it. If it glows that distinctive blue color, then you know that's a positive hit and should be reported to the river managers, water officials from the city, somebody that can come and do further testing to see if there is indeed a leak.
She says some groups have been able to pinpoint sewage leaks by deploying a whole series of tampons.
They put tampons every 50 meters or so along a stream, and they would get brighter as they got closer to the source of the leak. So they were able to pinpoint where the leak was happening because of the brightness of the tampons. And so you can actually put them systematically through a waterway to help identify the brightness and the location of leaks.
In addition to providing a low-cost method for communities to test their local streams, Albus says the project also gives young people a hands-on education on environmental monitoring.
It's a very, very great tool for youth to practice what it means to go and sample water on their stream. And I feel like it's almost like a gateway drug into water quality monitoring. You start with something simple like cotton on a string, and then you move all the way up to actually doing, E. coli sampling on your stream too. And it's a great entry point for some of the youth to get involved.
You'll find everything you need to know about tampling at SciStarter at this website. Well, that wraps it up for this episode. If you're tuning in via podcast and you don't have links to the projects mentioned, visit the SciStarter YouTube page where you'll find the video of this episode with all the project links in the show description. I'm Bob Hirshon. Thanks. This podcast is brought to you each month by SciStarter, where you will find thousands of citizen science projects, events, and tools and turtles. It's all at SciStarter.org. That's SciStarter.org. SciStarter's founder is Darlene Cavalier. And thanks so much to you, the listener and citizen scientist, for getting involved and making a difference. If you have any ideas you want to share with us, any things you want to hear on the podcast, you just want to say hi, get in touch with us at info@SciStarter.org. Once again, our email address is info@SciStarter.org. Thanks again, and I'll see you next time.